Greetings, viewers. I am Dr. Anukujo Uchenna, and I want to welcome you once again to my YouTube class. In today's class, we are going to be talking about something very, very important, something sacrosanct when it comes to the study of biology, and that is the X-raying of the cell structure. In this case, the animal cell. So the topic before us is cell structure and function. Now, of course, we know that the cell is the smallest and functional unit of a living organism. The way the atom is to chemistry is also the way the cell is to biology. The smallest unit and the smallest and functional unit of a living organism, and that is to say that life begins at the cellular level. Now, there are few other things that I would have loved to talk about before talking about the different cell organelles. I'll just mention just one or two of them, and that is the aspect of the cell theory. The cell theory is just a set of postulations by some scientists about the cell. And don't forget that the cell, which was discovered as a small room in oak tree, was discovered by the man called Robert Hooke in the year 1665. Now, scientists such as uh, Rudolf Virchow came and said that cells came from pre-existing cells. Cytoskeleton came and said that the body of plants is made up of cells. Then Theodosius K1 also came on board and he said the body of animals is made up of cells. These are the postulations that are being given when it comes to the cell theory. But like I said, my major emphasis in today's class is to look at some of these cellular organelles and the function that they perform. We're going to start with the nucleus. Now, when it comes to the nucleus, it discovered that, the nucleus was discovered by Robert Brown in the year 1831. And even though he discovered this nucleus, he didn't know the actual function that the nucleus performs. It was further research that made us understand that the nucleus is actually the cell government of the cell, meaning that the nucleus controls the activities of the cell. They contain a set of genes that help in the regulation of cellular activities. And as you know, the nucleus is at the center of the cell, where it helps to perform this function of regulation. It's like the headquarters of the cell. One of the specific functions of the nucleus is in what we call protein synthesis. Now, when it comes to protein synthesis, you will discover that every cell needs protein. Every cell requires protein, and it is the duty of the nucleus to help in this process. How does the nucleus do this? There is a genetic material that is called the deoxyribonucleic acid, the DNA, which is located within the nucleus. When protein is to be synthesized in the cell, what happens is that the DNA transcribes itself in form of messenger RNA and transfer RNA. Basically, we have other types of RNA. And the function of this messenger RNA and transfer RNA And of course, with ribosome, which is already found within the cytoplasm of the cell, the essence of or the function of these elements is to help in protein synthesis. And like I said, protein is important for every cell. For any cell that stops producing protein, that cell will begin to die. Some of the drugs we take, like the antibiotics drug, tetracycline, streptomycin, and the rest of them, one of the things they do is to go and stop the process of protein synthesis in organisms like bacteria. And once they cut off that process of protein synthesis, then the organism will die because protein is an essential part of life. So, in terms of protein synthesis, what happens is that the DNA transcribes itself in form of messenger RNA, and this is what we call transcription. So, within this DNA, the messenger RNA leaves and the transfer RNA leaves. So, once the messenger RNA leaves through the nuclear pore, messenger RNA and the transfer RNA. The transfer RNA goes to the amino acid pool. There is a pool within the a cell that is called the amino acid pool. That is where you have so many of the amino acids that are yet to be synthesized. So the function of the transfer RNA is to go to this amino acid pool to pick the amino acids. And once they pick the amino acid, they bring the amino acid within the ribosome. This is ribosome. And that is why it is said that the actual function of ribosome is that it is a site for protein synthesis. So the ribosome has about two different subunits, a eukaryotic cell, the 50S subunit, and the 30S subunit. So the messenger RNA comes to kind of line itself on the ribosome. So these are the ribosome, and this is the messenger RNA. So the transfer RNA, by the time it comes, then it begins to deposit the protein until a polypeptide is formed. So assuming the cell needs three amino acids like glycine, like asparagine, like phenylalanine, then the transfer RNA, according to the instruction of the DNA, goes to this amino acid pool and pick up these amino acids. There are other processes involved when it comes to protein synthesis, like proofreading and ensuring that the codon of the messenger RNA aligns with the anticodon of the transfer RNA. But I think I need to, have a special class where I specifically talk about protein synthesis. This class is all about talking about the functions of these different cellular organelles. So that is for protein, that is for nucleus and ribosome. They are involved in protein synthesis. Then we have the plasma membrane. When we talk about the plasma membrane, of course, that is the layer that shields the cell from external environment. So the one of the functions of this plasma membrane, which is also called the cell membrane, is that it helps in protecting the cell. But beyond that, it also controls what leaves and enters the cell, or have some molecules that are allowed to enter the cell, also have some molecules that are allowed to leave the cell. So it is a function of the plasma membrane to determine or to regulate what leaves and enters the cell. Then, of course, we'll have the cytoskeleton. When we talk about the cytoskeleton, we are talking about the, the skeleton of the cell. This is a structure that gives a mechanical support to the cell. And the cytoskeleton, just like the name implies, and just like I said, functions like the skeleton of the cell. We'll have different fibers that are made uh, from this cytoskeleton. We'll have the microtubules, we'll have the microfilaments, and we'll have the intermediate filaments. The microtubules, the microtubules, microtubules, microfilament, then intermediate filaments. Now, what are the functions of these fibers? Starting with the first one, which is the microtubule. The microtubule is one of the fibers made from the cytoskeleton, and the primary function is that it helps in movement around the cell. When you talk about movement, this movement could be either the beating of the flagella or the cilia. When it comes to amoeba, for the cilia to beat, for the flagella to beat, is because of the presence of these fibers, these microtubule fibers. Now, and apart from that, we also have vesicles that help in the transportation of molecules, or macromolecules like protein within the cell. For instance, in the Golgi bodies, when proteins are made or when proteins are sorted or packaged, they are being carried by vesicles into the cell membrane where they are needed. So anything that has to do with movement within the cell is being coordinated by the microtubule. Then when it comes to the microfilament, the actual function of the microfilament is to help in muscle contraction. Now, for muscles to contract, the primary fiber that helps in making sure that this is done or that this is achieved is the microfilament. And the third type of fiber is the intermediate filament. The intermediate filament helps in reinforcing the shape of a cell and making sure that some certain organelles, are the position of some certain organelles are maintained. The positions of some certain organelles are maintained. For instance, the nucleus. If you look at the nucleus is usually found at the center of the cell. So, in essence, this nucleus has some microfilaments that may not be easily seen except you use instruments that will help you see this. So these are fibers, and these fibers help to reinforce the position of the nucleus. If not, you can view a cell today and see the nucleus at the center. Then in the next few hours, when you view the cell, you see that the nucleus, is already here. That means the position is dangling. But the presence of these intermediate filaments will not allow this to happen. So that's the function of the cytoskeleton. Now, when it comes to rough ER, we have what is called the endoplasmic reticulum. And this endoplasmic reticulum uh, is of two types. We have the rough ER and the smooth ER. What makes ER to be rough and what makes ER to be smooth? The presence of ribosome on the ER is what makes it to be rough. And the absence of this ribosome is what makes it to be smooth. So we have in this case now, this ER that is attached to the nucleus is rough because you can see traces of ribosomes on the surface of this ER. And what's the function of the rough ER? The rough ER helps in the production of secretory protein, and it plays a role in the production of secretory protein. Example of this secretory protein is something like the antibodies. The antibodies are made in the rough ER, And once they make the rough ER, they are sorted and taken to the golgi apparatus where they are further sorted and processed before they are released into the cell to target foreign entities. That is the function of the rough ER. Then when it comes to smooth ER, the smooth ER helps in the synthesis of lipid. Then of course, apart from the synthesis of lipid, it also helps in catabolism, in metabolism of carbohydrates. That's the function of lipids and the function of carbohydrates. Function of lipid, function of the, what's it called again? Function of the smooth ER, I said lipid metabolism, then uh, no, lipid synthesis, sorry, that is production of lipid synthesis, then metabolism of carbohydrates, and of course detoxification. Yes, the smooth ER also helps in detoxification of toxins. Then another organelle is the peroxisome. The peroxisome is also a membrane-bound organelle, and it helps in lipid metabolism, mm, lipid metabolism, and enzyme catalysis. In fact, generally speaking, it helps in oxidation of some substances. That's the function of peroxisome. Peroxisome contains uh, enzymes such as catalase and oxidase. Now, some of the metabolic processes in the body brings about some byproducts such as hydrogen peroxide. And the function of the peroxisome and one of the enzyme it contains that is called catalase is to break this peroxisome, because peroxisome is harmful to the cell. So the catalase located within the peroxisome in breaking helps in breaking this peroxisome to form uh, water and oxygen. It breaks the peroxisome to form water and oxygen. This is the catalase performing this function. And don't forget, the catalase is located within the peroxisome. The structure in the body, or the organ in the body that is primarily responsible for detoxification is the liver. And that is why when you get to the liver, you see that we have an abundance of peroxisome where it helps in the detoxification of toxins or alcohol through this oxidation process. We also have the centroid. Now, when it comes to the centroid, the function of the centroid is simple, and the function is to help in cell division. Of course, you know that every cell divides. So the centroid helps in the production of the spindle fibers. If you can remember, the spindle fibers are usually produced during cell division. If you have this, so uh, the spindle fibers are usually produced, and that is to help uh, in the positioning of the chromosomes during this process of cell division. So the centroid helps in cell division, and the specific function is that it helps in producing the spindle fibers. Now, it is also important you know that this centroid is only found in the animal cell. You can't see centroid in the plant cell. And since you can't see centroid in the plant cell, the question now is how uh, if you can't see centroid in plant cell, sorry, yes, in plant cell. Now, how does or how do plants, or how do plant cells produce their spindle fibers in place of centroid? There is another structure in the plant cell that functions just like the centroid in producing the spindle fibers, and that is microtubule organizing centers. It is called the microtubule organizing centers. This is a structure in plant cell that helps in producing the spindle fibers. Then we have the lysosome. The lysosome is also a membrane-bound organelle that also helps in in digestion of debris or maybe say let me say molecules. For some organisms that feed through phagocytosis, like amoeba, he discovered that they kind of engulf food particles, and a membrane is formed within this food particle to form what is called a phagosome. Now, the lysosome may fuse with this phagosome to form another structure that is called a phagolysosome. That is when the lysosome now releases enzymes. It also contains, just like peroxisome, it also contains some set of enzymes that also help in the digestion of macromolecules. Enzymes like protease, enzymes like lipase, enzymes like nucleus. These are some of the categories or classes of enzymes found within the lysosome that helps in the digestion of macromolecules. Of course, you during the formation of a tadpole, for the tail, to be cut off and to be neutralized, the lysosome performs a function. Yes, in the metamorphosis of tadpole into frog, of course, tadpole has a kind of tail. Now, the lysosome helps in lysing that tail before it metamorphoses into the next stage of organism. Then, of course, even in the formation of human embryos, we were told or we know that humans have uh, webbed hand and feet, have some skin within this region. So, but the digestion of this skin is done with the help of lysosome. So you can see that lysosome has an important function that it performs. Then we have the cytosol. The cytosol is the semi-fluid part of the cytoplasm. It functions in cytoplasmic streaming, that helps in distribution of nutrients within the cell. And it is actually the center for some metabolic reactions, like glycolysis, for instance, which is one of the metabolic reactions, takes place within the cytosol. So it is the semi-fluid part of the cytoplasm where metabolic reactions occur and also helps in cytoplasmic streaming. Then desmosome, the point of attachment between one animal cell to another animal cell is what we call the desmosome. Cells are not in isolation. Cells are usually attached to each other. So that point of attachment between one animal cell to another animal cell is called desmosome. This will now lead us to differences between plant and animal cell. And I've mentioned some of them. Let me start with this desmosome I just mentioned now. When it comes to a plant cell, they don't have desmosome. They have what is called the plasmodesmata. Plasmodesmata. This is found in the plant cell, the point of attachment between one plant cell to another plant cell. But when it comes to animal cell, it's called desmosome. Another difference is that there is absence of centroid in plant cell, but is present in animal cell. Then, of course, what the structure I said that replaces centroid in plant cell is the microtubule organizing centers. Then, of course, I didn't mention the chloroplast, the chloroplast, energy transducers, the chloroplast, and the mitochondria. Now, the chloroplast is found only in plant cell; it's not found in animal cell. And that's why and of course, the chloroplast contains some pigments that help in harnessing the sun's energy during photosynthesis. Then when it comes to mitochondria, which is the last one, mitochondria, and don't forget that mitochondria and chloroplast are known as energy transducers. They transform energy from one form to the other. Now, This mitochondrion, this common name for mitochondrion is the powerhouse of the cell, and this is where energy is generated. This is an elaborate process, just like protein synthesis. So I'll just summarize it. So uh, when during the process of producing energy, glucose is broken down in the in the cytoplasm uh, through a process that's called glycolysis. Now, once this glucose is broken down into glycolysis, through a process called glycolysis into pyruvates, the pyruvates are now being taken into the mitochondrion. Now, within the mitochondrion, it goes through lots of processes, lots of processes like pyruvate oxidation, like Krebs cycle, and oxidative phosphorylation. Now, within the oxidative phosphorylation, that is when a phosphate molecule, a PI, is incorporated. This happens within the mitochondrion, is incorporated. And of course, we also have the ETC, the electron transport chain, through which in fact, that is even what happens before the final oxidative phosphorylation. There's an electron transport chain through which the electrons are being transported within the chain, and as they're being transported within the chain, they're releasing energy, and some of these energies will help in pumping some of the protons into the intermembrane space, and when it is pumped back into the matrix, it goes to activate the ATPs, which is ATP enzyme. And once the ATPs is activated, then of course it now helps in incorporating a phosphate molecule. This is ATP formed. So ADP plus PI now helps. That means the enzyme helps in incorporating a phosphate molecule into ADP, which is a raw material found in the cell, in the mitochondria. And once this is done, ATP is formed. And this ATP, the adenosine triphosphate, is now the energy currency of the cell. Once it is formed, it is taken back to the cytoplasm where it is used for metabolic activities. And as the energy is depleted, energy is being released. That means that as the sorry, phosphate molecules are being depleted, then energy is being released. Once it gets to ADP or sometimes AMP, that's adenosine monophosphate, it is now being transported back to the mitochondrion where the process occurs again. That is the summary of how energy is being produced in the mitochondrion. This is where I will stop for today's class, and subsequently, we'll talk about other aspects of this course. Thank you once more. I remain myself, Dr. Anukujon Uchenna.